In this video, we will discuss about drug efflux transporter to mediate is PDAC chemoresistance. In the previous video, we have discussed that when the pancreatic cancer organoid or single cells are kept in this high help matrix, then it will generate more chemoresistance against the drugs compared to the case where pancreatic cancer cells are in the organoid or single cell forms kept in this low help matrices. And we have discussed what this low help and high help matrices means in general, or in short. High help matrices means that it has high stiffness, and low help matrices means that it has low stiffness. And H represents the in the help. H represents the HA related proteins, and ELP is this elastic like proteins which are usually found in the tumor microenvironment of the pancreatic tumor cells. So let us begin to discuss the results that are presented in this figure. So in the panel A, first they need to, from the literature, they come to hypothesis that there are many drug efflux transporters are present in the pancreatic tumor cells. So they want to check the effect of this high help and compare it with the low help on this drug efflux transporter. So these are the drug efflux, some of the drug efflux transporter results are shown here. That is about ABCG2, ABCC3, ABCC4 and ABCC5. And what they have find out that in the case of high health pancreatic tumors grow in the case of high health has a high drug efflux transporter compared to the tumor cells that are developed in the low help. And same things in order to check whether this phenomena is only related to this drug efflux transporter or the other kind of proteins. So they also check this soluble liquid carrier or solute liquid. I'm not able to recall this SLC's full form, but they also check this family of SLC proteins. In this case, they check this SLC28A1, SLC28A3, SLC29A1, SLC29A2, and what they find out that the changes in this kind of proteins are either non-significant or are at minimum level. That can be seen here. Changes can be seen for this SLC28A3, SLC29A1, but not for this SLC28A1, SLC29A2, indicating that the effect of this high help matrices are more predominant on these drug efflux transporters. And these things, after after identifying this phenomena, they check this ABCG2, as this is more predominantly found in most of the cancer cells as a resistance protein. And what they have find out is that in the case of high help, the expression of this ABCG2 is higher compared to the low help. And these things they have also validated using the flow cytometry, that the high help has a more ABCG2 compared to the low help. And after this results, they wanted to check the effect. They wanted to check the effect of this drug efflux transporter. So they use the site population assay, where they use the hoist 33342 dye to check the efflux capacity of these cells. And the cells that have more efflux capacity will show low fluorescence, because they will also efflux out the hoist dye. And the representation is shown in this panel E. That is, in the case of help low, they are expecting that it should give more signals, and in the case of help high, it should give very less. It will give less signals, as less host dyes are accumulated inside the cells. And this is what they will observe in the panel F. That is, the red portions is their target of interest, which is the site population cells, as it will have low fluorescence. So when they check the results, they find out is that in the case of this help low, they see that low amount of cells are present in the site populations, whereas in the case of help high, high amount of cells are present in the site populations, indicating that more drugs efflux are present, more drugs efflux transporters, most drug efflux transporters are present in this help high tumor cells that are grow in this help high conditions, and when they knock down this when they knock down this ABC G2, what they have find out is that the site populations decreased both in the case of help low and in the case of help high, indicating that the results are mainly due to this ABCG2, and their comparisons are the quantitative analysis of this flow cytometry results are shown in this panel H, where they can see that the site populations goes down whenever they use the inhibitor for this ABCG2, indicating that the site populations are results because of the high expressions of this ABCG2 drug efflux transporter. And they check the results, and they check the results by sorting out the cell populations of these different by sorting out the cell populations of the uh, different group, that is uh, site populations and G0 G1 populations, and these populations are being separated out, and the other live cells, and they did the drug treatment using this gemcitabine, and what they have, and for control they use this DMSO, and what they have find out is that in the case of these site populations they see that more cells are grow compared to the case of G0 G1 when they compare it with the DMSO. In the case of viable cell also they see the reductions in the growth, and the quantitative analysis of this panel J is shown in this panel K, where we can see that site populations has more cell viability and is statistically significant compared to the case of G0 G1 and viable cells, where the cell viability goes down, indicating that these drug efflux transporters could be the reasons, as it will efflux out the these drugs, that is gemcitabine in this case, uh, and prevent these gemcitabines to act against these tumor cells. So I hope you got an idea how this drug efflux they come to the final conclusion from these results, that drug efflux transporter could mediate the chemoresistance, as they have shown in this figure with this ABCG2. If you need to understand these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.